So here we are looking at the three types of muscle tissue: skeletal muscle, cardiac muscle, and smooth muscle. One of the things you need to be able to do is to recognize the histology of muscle tissue. So essentially that just means be able to look at a photo of skeletal, cardiac, and smooth and be able to distinguish between the three types. So we're going to look at each one individually, and I'm going to point out some special characteristics that you need to be familiar with. So first up, we have skeletal muscle. Now, as far as physiology goes, remember, skeletal muscle is voluntary. It takes a nerve impulse coming from the brain to stimulate skeletal muscle to contract. It's located attached to bones, which is why we call it skeletal muscle, and it's very fast to contract. The minute the action potential or the nerve impulse reaches that neuromuscular junction at the muscle fiber, it's going to contract immediately. So it's very fast to contract. For physical appearance and what you need to look for recognizing skeletal muscle are a few things. First off, the fibers will be parallel with one another. Now, they can run in different directions, but all the fibers will typically be parallel with one another. So here in this photo, you're seeing the fibers running vertically. So you're seeing many pink fibers kind of all in line running vertically. It's possible for them to run transverse. They could run oblique. There's even sphincter muscles that have circular formations. So the direction can be in any direction. But understand that all the fibers will be running parallel with one another. Another characteristic to look for, and probably the key characteristic, is to look for those striations. Now, striations are the stripes that will appear. They're kind of tough to see because they're really thin and close together. But if you look toward the center of this photo, and you may even have to pull your iPad or your phone or your computer, whatever you're looking at, like really close and kind of zoom in and look at this. But you can see some stripes. I'm going to kind of draw a bracket in some of the areas where it's a little bit darker, but right in here in this area you can really see some stripes, some striations in there. They're really thin, but it goes light dark, light dark, light dark, light dark with those stripes. This is the I band, which is the light band, and the A band, which is the dark band. And it's the arrangement of those myofilaments, your actin and your myosin, that's creating those striations. So when you're identifying skeletal muscle, look for fibers running parallel with one another and look for those striations. Now here we're looking at cardiac muscle tissue. Now, cardiac muscle tissue does share some similarities with skeletal muscle. First, it is striated. Now, in this photo, those striations are really tough to see, but again, if you were to move really close to the screen and look closely, you can see a few stripes. This is an area right in here where I'm able to see a few striations in there. And again, it's the arrangement of those actin and myosin filaments in there. There are differences, however, between cardiac and skeletal muscle. The striations are pretty much the only thing they have in common, other than the physiology working very similarly there. But physically looking at it, you can clearly see that the fibers of cardiac muscle tissue are much more branched. They're not near as parallel as those skeletal muscle fibers were. You've got some spaces between those fibers, so they can start to branch off in different directions. The fibers are also multinucleated there, and probably a key characteristic, something that you really need to look for and hone in on when you're identifying cardiac muscle tissue, is the presence of an intercalated disc or multiple intercalated discs. Intercalated discs are thickenings of the sarcolemma where two adjacent cardiac muscle tissues attach. It's pretty much a junction where you have two fibers connecting with one another. In the intercalated disc, you have desmosomes, which are the junctions that hold them together during contractile motions, and you have gap junctions. Gap junctions allow the ions to seamlessly transfer from one fiber into another fiber and keep that contraction process moving smoothly. When cardiac muscle tissue contracts, it contracts automatically. So we describe that as being autorhythmic. So it exhibits autorhythmicity. It contracts almost on its own, okay? So having these junctions allows the electrolytes, or the ions, and in particularly sodium and potassium, to easily and seamlessly transfer from one fiber into another fiber. So I'm going to point out a few intercalated discs on this diagram. You do need to be able to recognize these. So right here is a really good intercalated disc. They will run transversely to the muscle fiber. Okay, so I'm going to label it intercalated disc. Okay, intercalated disc. So there is one. Here is one. They're all over the place in this diagram. I'm just going to pick out a few. Here is another one right there. Here is one here. I mean, they are connecting all your fibers. So they're very prominent. They're all over the place. I mean, I can go through and I can circle a ton of them all through these fibers within that. So that's a key characteristic that sets cardiac muscle apart from skeletal muscle. Okay, so it's branched, those intercalated discs are present, and of course location. Cardiac muscle tissue is only found in the heart, so it's specific to that location. And don't forget, for physiology, when it contracts automatically, so it exhibits this autorhythmicity. So now we have smooth muscle tissue. Now smooth muscle tissue gets its name because it has no striations in it. So literally it's smooth. It looks very similar to connective tissue where you just have a lot of collagen fibers running everywhere, because smooth muscle tissue will appear like these smooth pink fibers. But now it's not connective. It's muscle tissue. Now the cells, or the fibers of muscle tissue, of smooth muscle specifically, have this spindle shape to them. They tend to taper off at the ends. So I'm going to outline a few of these cells in this photo so that you can see what I mean by that spindle shape. So right here actually is a smooth muscle fiber. So you can see it kind of comes to a point here and then narrows and comes to a point there. That is a smooth muscle fiber. Another one here, kind of outline that. There's another one right beside it, right here. Kind of pull these together. So I'm trying to outline these for you, just so you can kind of get a visual of the fibers themselves. So I've outlined several smooth muscle fibers. You can see how they kind of taper off at the end. So a wider belly in the middle, and then they taper at the end. And you can come up to the screen as close as you want to. There's no stripes in there, so no striations. That's why we tend to call it smooth muscle. Smooth muscle tissue is found in hollow organs. It's also sometimes called visceral muscle tissue, since it's found within organs. So like your stomach that churns, your intestines that wiggle and writhe during digestive activities, your blood vessels that can vasoconstrict and vasodilate, the urinary bladder that can contract and relax in women, the uterus in females there for childbirth and uterine contractions. All of that is smooth muscle tissue. So hollow organs contain this smooth muscle tissue. It is involuntary. There's a single nucleus per cell. And then if we compare the contraction process to cardiac and skeletal, it's actually quite slow to contract compared to the others. Skeletal is fast to contract, cardiac muscle is automatic, and then we say that smooth muscle tissue is slow to contract. So now we've come full circle and we're right back to where we were when we started. So we have our three main muscle types here. Skeletal muscle, look for those striations and those parallel fibers. Cardiac muscle tissue, look for more branching in the structure and find those intercalated discs. And for smooth muscle, remember, no striations, the cells taper at the ends. Smooth muscle tissue, smooth, no striations there. Be able to identify these three muscle types given a photo on your test and on your lab assignments.